Now, regardless of which membrane we're talking about, all membranes will be made up of a phospholipid bilayer. If you need to review the structure of the phospholipids in Chapter 5, go back and look at that. But keep in mind that we're going to have that little glycerol backbone that will have two fatty acids coming off of it, and then in the third position we're going to have our phosphate group. What this means is that we end up with fatty acid tails here, which are going to be hydrocarbon tails, or they're going to be nonpolar, or we could also use the term hydrophobic. And then where the phosphate group is, up at the top of the phospholipid, we're going to have a hydrophilic area. So we've got one end of our molecule that's hydrophobic and one end that's hydrophilic, and because of that we do call it an amphipathic molecule. When we're talking about the bilayer that's going to make up a cell's membrane, it's two layers of phospholipids, and they're oriented so that all of those fatty acid tails are on the interior or the inside of the membrane, which means that the inside of the membrane is certainly going to be hydrophobic. We could also call the interior of the membrane nonpolar, because those are nonpolar covalent bonds that make up those fatty acid tails. On the outsides of the membrane is where we're going to have the hydrophilic head groups, and those are the areas that would be exposed to water. The phospholipids are making up the structural framework of the membrane, but they're not really giving the membrane its unique functions. So that's where proteins come into play. Associated with the membrane's phospholipids, you will have lots of proteins. These proteins are going to be either loosely attached to one side or the other of the membrane, or they may be completely inserted in the membrane. They may span one side of the membrane all the way to the other side. Now, it is important to remember that hydrophobic things want to be next to other hydrophobic things, or nonpolar wants to be next to nonpolar. So if you do have a protein that is spanning the membrane, as we have right here, this protein is going to have different areas. On the inside, it's going to be nonpolar on the outside here. This is because it is touching nonpolar stuff. So all of this area of the protein that's touching the nonpolar groups is also going to be nonpolar. However, up here on the top, it's exposed to polar things. So that part of the protein is going to be polar on the outside. If we're talking about proteins that are loosely attached, we would expect that these would be polar on the outside because they're in the presence of water. So keep that in mind when we're talking about membrane proteins. They must match whatever they happen to be touching. Otherwise, they're not going to insert in that membrane whatsoever. The phospholipids of the membrane are really just held together because of the desire for nonpolar groups to be next to other nonpolar groups. So we call this hydrophobic interactions. Just like if you take a bunch of oil and you throw it in a glass of water, that oil automatically comes together or associates with each other in one big drop. And that's going to minimize its exposure to the water. In the same fashion, if you throw a bunch of phospholipids into a glass of water, they automatically assemble so that all of the hydrophobic tails are next to each other and the hydrophilic part is next to the water. And that's really how we come about getting that phospholipid bilayer. This is important because if we don't have covalent bonds between the phospholipids, this means that they are free to move about. So they will certainly be moving a lot in the lateral direction, and this means within the same layer. And if you have proteins inserted, they're going to be able to move laterally as well. What we don't have a whole lot of is movement from one layer to the other layer, or what we call the flip-flop. Now the reason for that would be that if we were to get the flip-flop, we would have to take, say, the hydrophilic group. It would have to pass through the hydrophobic core in order to get to the other side. We said hydrophilic things would much rather be next to other hydrophilic things. And the hydrophobic core is excluding hydrophilic things from passing through as well. So this is going to only happen on rare occasion. It does happen, but not a whole lot. This also helps to emphasize the type of molecules that are going to have an easy time moving through that membrane. If hydrophobic things like to go next to hydrophobic things, if we're talking about molecules that are going to pass right through the membrane, they're going to need to first of all be small so that they can squeeze through, and then they're also going to need to be nonpolar. So if we have both of those characteristics met, then they would be able to pass across that membrane without any difficulty. A good example of this would be oxygen gas, or CO2. It's a small molecule, it's nonpolar, it can easily pass through there. If we're talking about other small things, like an ion would have a lot of difficulty getting through that hydrophobic core because it has a charge. And charged things are hydrophilic. Keep in mind they're not hydrophobic. We'll certainly need to move ions into the cell, and we'll have to have special mechanisms to help that happen because they're not going to get through on their own. That's where this selective nature of the membrane comes into play. The fluidity of the membrane is very important. So fluidity means the movable nature of the membrane. So we're talking about liquid versus solid here. Keep in mind that the membrane is made up of phospholipids that have two fatty acid tails on them. We previously talked about the fatty acid tails, or fatty acids, and how they were affected by temperature. We said as the temperature goes up, they're going to melt or become more liquid-like, and as the temperature decreases, they would become more solid. Keep in mind that the degree of saturation affected that as well. So if we had something that was saturated, we talked about our saturated fatty acids, those were solids at room temperature. And if we talked about our unsaturated fatty acids, those were liquids at room temperature. So essentially, we have this balance that must be maintained in the degree of fluidity of a membrane. The higher the temp, the higher the fluidity. And then in the opposite way, the decreased temp, the lower the temp, the decreased fluidity. If we talk about our tails, basically the more unsaturated, then the increased fluidity. And we could say the more saturated, it's going to have a decreased fluidity. So we want to understand what's going on here. If it is warm outside, then the cells can very well have a lot of saturated fatty acid tails. Because if it's warm outside, those fatty acids are not going to get solid in nature. However, if it is cold outside, then we would need to have more of what we see on this side of the slide. Because if it's cold outside, we don't want the membrane to get solid. We want to incorporate some unsaturated fatty acid tails to help spread the molecules out so that they stay fluid. Cells are very dependent on the movable nature of their membranes. If their membranes ever get solid to where molecules cannot move around within them, then that is deadly to a cell. So cells are able to adjust their membrane composition according to environmental conditions. However, some cells are a lot better at that than others. We think about plants. Plants are in specific locations. They cannot change their environment when it is cold outside or when it's winter time versus when it's summertime. They're in the same place. So they have to adjust to whatever that temperature happens to be. If we're talking about winter time, we know that the temperatures get low and many plants are damaged by those low temperatures. If we talk about what we typically call chilling temperatures are cold, but they're not freezing. And there are some plants that are damaged or even die when the temperatures don't even get down to the freezing level. The problem with those plants, and these are going to be your tropical plants, the problem with those is that they're unable to make this adjustment that we see right here. So their membranes are typically a lot of saturated fatty acids. So this means that that's fine when the temperature is hot outside, but if the temperature becomes cool, it's going to turn solid. So they would need to incorporate a lot of unsaturated fatty acid tails, but they're not able to do that. So for those, they do get damaged or even die when we have low temperatures because their membranes become solid. So it has absolutely nothing to do with ice formation in that case because the temperatures haven't got that low. It is important that you understand the correlation between temperature and how fluid the membrane is, whether it's very fluid or solid. And then we also want to understand how the fatty acid tails affect that. We also don't want the membrane to be too fluid because if it's too fluid, it's completely melted and now we're not really getting that phospholipid bilayer around the outside. We're then going to have essentially everything falling out of the cell, which would certainly be deadly as well. Another thing that does affect the fluidity of the membrane is any other molecules that you happen to have inserted. So that would include steroids as well, because those are going to be hydrophobic and they can insert in that membrane. So we've talked about the phospholipids that are going to make up the bilayer of the membrane. We've talked about the proteins that are going to be inserted in there as well. So if we just look at those proteins in a little more detail, those proteins a lot of times are going to be integral proteins. Integral proteins is just referring to proteins that touch, even if just a little bit, that hydrophobic core of the bilayer. They may be just partially inserting into the hydrophobic layer. They may be completely spanning it. So these, if they're running from one side of the membrane to the other, we call these transmembrane proteins. The transmembrane proteins, because they're kind of exiting on both sides of the membrane, a lot of times they can tell the inside of the cell what's going on outside the membrane, or they can help to move molecules across that membrane. In addition to the integral proteins, we also have the peripheral proteins, and the peripheral proteins would be the ones that don't touch the hydrophobic layer at all. So generally speaking, these are going to be hydrophilic or polar on the outside, and they would just be loosely kind of attached. The proteins are considered to be the functional part of the membrane, so they do give membranes their specific characteristics. This is what makes the plasma membrane different from, say, the nuclear envelope or the vesicle membrane. And if we look at some of those functions that proteins can take on, one that we'll talk about a lot in this chapter is going to be the transport function. While small nonpolar molecules can move across the membrane just fine, other molecules cannot. So we'll have to have help to get them across, and we will have proteins involved in that help. There will be some proteins in the membranes that serve as enzymes. We'll look at those a bit in chapters 9 and 10. Some molecules are involved in signal transduction. So these are the ones that can basically bind a molecule on one side of the membrane and then change shape in a way that it tells the inside of the cell what's taking place on the outside. Some of them will be involved in recognizing other cells, attaching cells together, and then just holding everything together in general. So this may mean holding the cytoskeleton to the plasma membrane and then also holding that extracellular matrix to the other side, if we're talking about an animal cell. In addition to the phospholipids and the proteins, we also have carbohydrates associated with the membrane. These carbohydrates are generally speaking going to be rather short, so less than 15 units long. Technically, they would be called polysaccharides, but polysaccharides are usually quite a bit longer. Usually we're talking thousands with polysaccharides. These membrane carbohydrates can be bound to lipids, in which case we call them glycolipids, or they can be bound to proteins, in which case we call them glycoproteins. Notice in this slide, the carbohydrates are the green parts, and notice that if we're looking at the plasma membrane, as we are in this picture, that they're all in the outside or the exterior of the cell. This is important because they will be responsible for interacting with the surface molecules of other cells and thus facilitating cell-to-cell recognition. What this means is that each cell type has specific carbohydrates that it has on the outside. This helps cells to recognize each other or to distinguish a foreign cell. If you had a foreign cell, then the cells would recognize it as that, and then they could mount, say, an attack against it or an immune response against that particular cell. So very important when the membrane is constructed that those carbohydrates do get put on the outside of the cell, because if they're not on the outside, if they are pointing towards the inside, they will never be detected by the neighboring cells. So membranes do have distinct sides or faces to them. So the inside of the membrane is definitely going to be different in composition than the outside of the membrane. And we just pointed out that the carbohydrates would be on the outside of the membrane. This does affect the way that the proteins are synthesized in the endomembrane system. So it does go back to the secretory protein synthesis in the rough ER, and then the modification in the Golgi apparatus. Keep in mind that once the proteins are inserted in the membrane, they cannot be turned around, because that would be the flip-flop, and that would be very difficult because that will require a large hydrophilic portion to pass through that hydrophobic core. If we look at this picture, which does go back to chapter 6, up here in the ER we would have the synthesis of this particular protein, which happens to be a transmembrane protein. So notice that when it is first synthesized by ribosomes stuck on the rough ER, it is going to get inserted in the membrane. And that is the orientation that it will always be in. So it must be inserted correctly the first time. On here we have this little kind of U-shaped structure. That's going to represent a carbohydrate. Notice it is on the inside of the membrane. If we pinch that vesicle off, here we can see that the U part is still on the inside of the membrane, and we can dump it into the Golgi. It's still going to be on the inside, and then we're going to send that ultimately to the plasma membrane, which will fuse, dump the contents, and then we can pull it tight. When that happens, notice that your carbohydrate is going to be located on the outside of the cell, which is exactly where we want it to be. It's not a mistake that it ended up like that. The cell knows essentially that the inside of the ER, Golgi, or vacuoles and vesicles will end up outside the cell. It pays attention to that, and when those proteins are initially inserted in the membrane way back in the ER, they are inserted in the proper orientation to make sure that whatever is supposed to end up on the outside of the cell or outside of the plasma membrane will, and whatever is supposed to end up on the inside will end up on the inside. If it doesn't happen like that, then we have essentially wasted our time, and we would have to completely remake that particular protein.